بسم الله الرحمن الرحيم والصلاة والسلام على أشرف الأنبياء والمرسلين نبينا محمد وعلى آله وصحبه أجمعين. My name is Abdullah Abbas Al-Ghamdi, and today inshallah I with my group we will talk about the determination of carbohydrates in honey sample by ion chromatography mass spectrometry. So let's start with honey. Honey is a natural food with very high nutritional value. It is formed by bees from the nectar of the planet flowers. It is a complex mixture of many compounds such as carbohydrates, including all its classes, and I mean here monosaccharides like fructose and glucose, which make 65 to 85 of the sugars in honey, and also disaccharides and oligosaccharides. It contains 13 to 18 type of oligosaccharides, which is important for determining both the geographical and botanical origin of honey. And it also contains vitamins such as vitamins B and vitamin C. And it has many benefits and uses. For example, it's considered as neutral sweetener, and it's used as antibacterial, and it's used for treatment of many diseases. So why do we do this analysis? And what is our purpose of this analysis process? There are many reasons for analyzing a sample of honey, and each study must talk about specific reason. For example, there are studies to determine the amount of carbohydrates in the sample of honey, and other studies to analyze the validity of its use and the preservation material that are found in it. But the study that we conducting our research on was aimed at determining the type of carbohydrates in honey sample, especially with oligosaccharides, because they are used to determine the origin of the flower, and this help to know the geographical and botanical origin of honey. Let's talk about the history of the honey analysis. Since ancient times, humans have found many ways to analyze honey for many reasons, including testing the viscosity by stirring or the sweetness by testing. But with development of science and development of the goals of honey analysis, many methods of analysis have been conducting, including HPLC with UV detection to determine the some oligosaccharides, and also gas chromatography-mass spectrometry, which consider the most common method of analysis for honey, and also gas chromatography with the flame detector. But all of these methods was time consuming and required a lot of substance and materials, until they use HPAEC-MS spectrometry. They find it more powerful for analytical tool and high sensitive and selective, and this is what makes it so great to use. And that's all about my part. Thank you very much for your listening.
Assalamu alaikum warahmatullah. My name is Abdulaziz Sabr, and today I'm going to talk about the high pressure anion exchange chromatography that is coupled to mass spectrometry analytical method and how it does work. The first thing we have to know that it's a two-steps process. The first step is the high pressure anion exchange chromatography. It provides a valuable and powerful analytical tool, and its main function is that it separates the sugars in the honey sample. So it is mainly used for the separation. Why? Because many carbohydrates present slight acidity at the pKa between 12 to 14. Another condition is that in alkaline condition, the hydroxyl groups are converted into oxyanions, making it possible to selectively separate, which is what makes this method special. That it's you can be selective in the species in the anionic form. The third thing is that the separation can be strongly influenced by the number of hydroxyl groups present in the compound, by their position inside the sugar, and by the degree of polymerization. For this is what concerns the first step, which is the high pressure anion exchange chromatography. Moving on to the second step and the role of the second step, which is the mass chromatography. First of all, in the mass chromatography, we're going to discriminate between the type of saccharide, the monosaccharides, disaccharides, trisaccharides, and the tetrasaccharides. This will lead us to reduce the number of peaks in each ion chromatogram. Moving on to the next point, we have to know that the linearity of the calibration curve of each sugar was estimated using a series of standard solution of the sugars at average concentrations 0.05, 0.25, 0.51, and 2 and 4 milligrams per liter to the power of minus 1. What we know here is that these numbers are the numbers that were used to compare the results between the standard solutions. This one is used to compare between the graph that we will get and the concentrations we have. Moving on to the next step, which is sampling. Sampling is nothing but how and where we get our samples from. The first thing is that these honey samples of various botanical origins were directly collected from the Apiarists Association and in the farms of beekeepers. The second thing is that there was the use of different geographical areas of the Trentino Alto Adige region in Italy and harvested between 2017 and 2018. Also, two commercial Argentinian samples were also analyzed just for the sake of comparison. The third thing is that there is a total of 43 honeys with different floral origin that were used in the study. Moving on to the sample preparation and the steps we have to do in order to prepare our sample. The first thing is mixing of the honey sample to guarantee homogenization and that to make the whole solution homogeneous. The second thing is that the pre-softening of the crystallized honeys. We do that by heating in a thermostatic bath at 40 degrees Celsius. The third thing is that each sample is weighted 50 mg in a 50 ml volumetric flask, and in every sample is spiked with 13C6 glucose as internal standard. And the fifth step is that the specimen is going to be diluted with ultra pure water in a volumetric flask until a final concentration of 0.1 mg in each milliliter to the power of minus 1. We do this dilution because it allows to reduce the glucose and fructose concentration, which is going to be very beneficial when it comes to the chromatogram.
Assalamu alaikum wa rahmatullahi wa barakatuh. I'm Majid Khan. I'm today going to talk about the unique contribution to science. So, as we could see, the procedure developed a fast, simple, and inexpensive, without any expensive steps, such as purification or the solvent extraction, which also reduced the sample preparation time from using a solvent-free, ultra-pure water that been required in this method. And the chromatographic separation, coupled to the mass spectrometer, only required one to separate the isotopic species, providing a better peak resolution in comparison to the other detector, where all the saccharides are determined in one single chromatogram. The sugar's peaks were all resolved, and an accurate quantification chromatographic run has been developed in this method. As we could see in this slide or in this diagram, that all the peaks are accurately measured and shown in this diagram. I'm going to continue here talking about the summary. Honey is a natural food which contains a percentage of 65 to 85% of it is glucose and fructose mainly, and a diverse types of oligosaccharides, which are important in determining the geographical and phytic organs of the honey. And it also contributes to many benefit uses, such as it's to be an antibacterial, or that is a sweet compound in nature or in the wild. And the purpose of the analysis process is to characterize honey of different floral organs and determine both the geographical and phytic organs of the honey. by using the high pressure anion exchange chromatography coupled to the mass spectrometer. And the analysis process focused on sampling 43 types of honey that have been collected from the apparatus association and in the farm of the beekeepers, with different follower organ, which is used in sampling preparation that lead to an expensive and time-saving method. And thank you for listening to our presentation. And here are references that we use it in here. And thank you again.